I'm gonna just give a really brief talk about my path to graduate school and the decisions and factors that I weighed in choosing a graduate program. So my name is Caitlyn Fastenau. I'm a minority student I was a minority student at the University of California, Davis, where I began my journey in science. So I entered UC Davis as a freshman in 2016, where I agreed to join or was accepted to join a biological undergraduate scholars program, which then led into my advancement into ADAR and ADAR honors throughout my undergraduate experience, where I joined a lab that focused in cognitive neuroscience. So I worked mostly with patients. We did cognitive assessments, neuropsychiatric assessments, and produced data looking at the positivity effects in older adults and retaining memory and cognition. I really enjoyed my time working with patients, and I thought that was like the path that I would take throughout graduate school. As I got more into my undergrad and some basic science courses, I found that I really wanted to find research that bridged the bench and patient-derived research. So I then did my summer externship in the summer between my junior and senior year at the Buck Institute for Research and Aging. I was the first student from UC Davis ADAR to start that sort of collaboration with the Buck, which has continued after me, which I was really excited to spearhead that. I worked under Dr. Tara Tracy using a transgenic mouse model of Alzheimer's disease focused in tauopathy, and we worked also with iPSC-derived neurons. I absolutely loved my experience with that externship, which pushed my interest into applying to graduate school in the next fall. When I was looking to apply for graduate school, I knew I had a vested interest in aging research and wanted to continue in a specifically aging-focused graduate program. Unbeknownst to me, there aren't as many as one would think that have a very strong and, I guess, pinpointed aging program. And so there are only about 5 in the country that were really sparking my interest. And one of those programs is the UT Health San Antonio Biology of Aging program. So I'll speak a little bit more about that in a minute. When I was choosing a graduate program, I graduated in the middle of a pandemic. So there were a lot of factors that kind of played into my decision of attending graduate school. And one of those things is obviously you need to think about how graduate school will benefit your career, but also what research fields you'd be interested in pursuing. Again, location is super important. I knew that I didn't want to live anywhere cold. I'm a California born and raised and could not handle the snow or really any weather. So I decided to apply to programs that only had more of a warm climate, but also applied with my interests. And that's kind of how I ended up in Texas. Again, also you have to consider your type of learning experience that what you want and what kind of, for example, TA experience. I go to a graduate school that doesn't have an undergraduate affiliate, so there's no requirement for TAing, and our stipend is fairly substantial for the cost of living in the state I'm in, which is Texas in the city of San Antonio. So that was a major proponent of also why I wanted to come here. So to talk a little bit about UT Health, as of, I believe, May of this year, we became a National Institute on Aging designated Alzheimer's disease research center, which was spearheaded by one of the labs that I work in, and honestly is a massive advantage for this institution. Additionally, this institution has some of the best clinical and cores that are offered for aging research. We have the Biggs Institute for Alzheimer's and Neurodegenerative Diseases, which both of the labs that I work in are heavily integrated into that center, where we do both basic science research and clinical research. We also have the Barshop Institute for Longevity and Aging that do interventional research like caloric restrictions and different types of cohort studies in a variety of animal models. We also have the Pepper Center for Interventional Medicine, which looks at longitudinal interventions for human patients. I particularly chose to join the discipline of biology of aging, which really is a great discipline in my opinion. I'm a little biased, but we basically focus in molecular, cellular, and physiological geroscience, and we're trained extensively in basic science mechanisms of disease. So the main difference in this program compared to other programs is everything we do is disease oriented in some fashion, which was my driving force of wanting to do a program that was disease oriented. So now I'm going to talk a little bit about the research that I've been doing. I'm a second-year graduate student, and I've been focused primarily on one of the immune cells of the brain called microglia. So to talk about my project, I have this little schematic that I've developed, and basically it's complicated, but I'll try to walk through it as best as I can. So I'm really interested in a post-translational modification called sialylation. And in this modification, we know it's present in microglia. Again, my immune cell of favor. So here's a little diagram of a homeostatic microglia. The little red balls are what I'm kind of describing as sialic acid modifications. And there's an enzyme inside the lysosome of the cell body called NU1, and those interact with the cell surface sialic acid. When activated, NU1 translocates to the cell surface and is able to desialate both post-translational modifications as well as interacting receptors. In that process, I'm hypothesizing that leads to activation of microglia based on cleavage of those sialic acid modifications and allows microglia to interact with amyloid beta plaque pathology in Alzheimer's disease. And we know activated microglia play a massive role in clearing debris as well as compacting toxic A beta plaques into cord A beta plaques. So in order to investigate this more thoroughly, I did a deep dive into literature and found that there's very little visual representation of this post-translational modification in Alzheimer's, and specifically the localization of this post-translational modification. Therefore, I have primarily been focusing over the past 6 months on visualizing what this might look like. So we're employing our 5xFAD transgenic mouse model of Alzheimer's disease. And here I've been doing some immunoblotting and some pretty creative carbohydrate staining to identify and visualize sialic acid modifications on microglia. On the left side is our transgenic mouse brain, about 10.5 months old. I've isolated the cortex region of the brain, and I've used immunohistochemistry and IS to stain for IBA1, which is microglia, and our post-translational modification of sialic acid with DAPI. And as you can see, there are green puncta co-localizing with the red IBA1 stain, visually showing us that we can see sialic acid modification on the cell surface of microglia, which is really the first time it's been visualized. And because I work with a lab that's directly associated with the brain bank of UT Health, which basically has amazing postmortem human tissue at our fingertips to work with, I was able to obtain postmortem human AD tissue and kind of replicate the stain. And I'm seeing the same phenotype that I'm seeing in our mouse, which is one, validating our model, and two, showing us that this might be a very clinically relevant topic to focus on because we can see it in postmortem tissue. So right now I'm working on more of a mechanistic approach to understand the enzyme interactions and how that might be manipulated in disease and exacerbated in certain conditions. So that's kind of my brief overview of what I've been doing here. I just want to highlight that this institution is fantastic. There's great funding. There's a strong representation in biology of aging and in aging faculty, and it's a sought after institution for those that are interested in aging. Here's the labs and the centers that I'm involved with. We had an amazing undergrad that drew pictures of our lab, so here we are. And nothing happens in science in a bubble, and working with a great team and a great set of labs is really crucial. And I've absolutely enjoyed my time here thus far.
Thank you. Thank you.